So you understand the basics of genetics, and now we're going to talk a little bit about Gregor Mendel and how he actually figured out how traits pass from one generation to the next by using pea plants. So modern genetics began in the mid-1800s in an abbey where a monk named Gregor Mendel documented the inheritance of peas. The reason we talk about experimental design at the beginning of this year is because these experiments can actually mean something. Gregor Mendel actually used a very good experimental design, and he did a mathematical analysis, which just means that he went and collected data and then kept records of them. So this is an excellent example of the scientific method. So what he actually did, he cross-pollinated true breeding parents, and these parents, we're talking about pea plant flowers. He raised the seed and then observed the traits. So here we see the cross-pollination. He crossed a white flower with a purple flower. He raised the seeds and he got all purple flowers. He allowed the offspring to self-pollinate and observe the next generation. And all that means is he let two of these offspring fertilize each other and then observed what kind of seeds actually resulted. So Mendel collected data for 7 traits of the pea plants. So here he saw the purple versus a white flower, a yellow versus a green seed, and then a round versus a wrinkled seed, green pods versus yellow pods, inflated pods or constricted pods, axil flowers or terminal flowers, and hall plants or dwarf plants. And the axil flowers and the terminal flowers, if you look at the pictures, the axil flowers, the flowers actually coming from like the middle of the stem, while the terminal flowers, terminal generally means the end, so the flowers are actually coming from the top of the plant. If you keep looking at this table, you will see that he found out that everything on the left was a dominant trait, while everything on the right was a recessive trait. This F2 generation, all the way to the right, the F2 generation just means that it is an offspring generation, and we will talk about that in a minute. But you can see the ratios between dominant and recessive. So looking closer at Mendel's work, the parents were a true breeding purple flower with a true breeding white flower. The first generation were hybrids of the two, and they all expressed the purple color. They self-pollinated and resulted in the second generation. The second generation showed that 75%, or 3 out of the 4, were purple, while 1 out of the 4, or 25%, were white. So, to distinguish between these generations, you can shorten those words to P1 for the parent generation, F1 for the first generation, and F2 for the second, and so on. So what did these findings actually mean? Well, he figured out that some traits can mask or suppress others. So purple and white flower colors are separate traits that do not blend. So we talked about this in class, and this was a dominant allele. Versus a recessive allele. Alright, so this goes into a little bit of detail about the alleles and how they actually function as a part of DNA. So a dominant allele is basically a functional form of the protein, and it will affect the trait. A recessive allele is basically a non-functioning protein and has no noticeable effect on that trait, which is why a dominant allele is what is depicted in the phenotype, while a recessive allele is not. Alright, so what was his conclusion? Well, biological inheritance, so traits that are passed from generation to generation, are determined by factors that are passed from one to the next. Today, we call those factors genes. So genes can be transferred from one generation to the next and determine the traits that you get. Mendel came up with two laws, the law of dominance, the law of independent assortment. For the law of dominance, some factors are able to mask others. In other words, some alleles are dominant and others are recessive. The law of independent assortment deals with meiosis. So when gametes are formed, the two alleles are segregated so that each gamete carries only a single copy of the gene. So we did talk about this in class. I think y'all understand it. The difference between how an organism looks and what its genes are actually coded for. The phenotype is the physical description of the organism, while the genotype is the description of the genetic makeup. So here in this picture, it's asking us to explain Mendel's results using dominant and recessive phenotype versus genotype. So here, the purple, when you cross a purple with a white flower, ended up with a purple flower in that first generation. Purple was the dominant trait while white was the recessive. You can see that because when you crossed the two, you ended up with purple and not a mixture or a white flower. So the phenotype versus genotype, while this is a purple flower, the genotype may be heterozygous, so it may have one dominant and one recessive allele. These alleles can be represented as letters. So for this case, we have flower color alleles is either big P or little p. True breeding purple flowers are dominant. They are homozygous dominant, meaning they have two big P's. True breeding white flowers are recessive, so they would be homozygous recessive or have two little p's. So in our example, the parent generation would have a purple crossing with a white flower. So we would have a homozygous dominant crossing with a homozygous recessive. In that first generation, all of them would end up being heterozygous with one big P and one little p. So if we put these in a Punnett square, you get this. You put the male on the top, the female on the bottom, and if you cross two heterozygous F1 plants, you get this. So here we've taken the male big P with the female big P and you get a heterozygous. Here and here you have one big P or one little p, and these are both heterozygous. And then here you have two little p's, one from the male, one from the female. So in the top left part, you end up with a purple flower. In both the heterozygous, you get a purple flower. And in the fourth, the homozygous recessive, you get a white flower. So here you can actually distinguish between genotype and phenotype. In genotype, it is 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. To compare that with phenotype, you will have 75% purple and then 25% white. These ratios are important because they can actually tell you what the parents are. So Mendel worked with a very simple system, and P's are genetically simple. Most traits are controlled by a single gene. He got lucky in this. Many things do not work like this. So each gene in the pea plant only had two versions. So a completely dominant, a big A, or a recessive, a little a. However, what happens when it does not work this way? Well, you can have several things happen. You can have incomplete dominance, where it creates hybrids that have an in-between appearance. So, for example, in carnations, if it is homozygous dominant for the color red, you'll get a red flower. If you are homozygous recessive, then you'll be white. But if you are heterozygous, meaning you have one big R and one little r, then you end up being pink. That's because that dominant R makes 50% less color than if it was either big R, or it had no color with little r. So these we can actually write using two different letters because it is showing two different phenotypic traits. So we can say that the red flowers are big R, white flowers, true breeding, would be big W, and pink flowers would be the mixture of the two with a big R and a big W. So here you can see true breeding red flowers, which would be big R, with true breeding white flowers would be WW. That first generation ends up being a combination of both, meaning that they are pink. And then that F2 generation is when these ratios start coming into play. So here it's a 1 to 2 to 1, meaning 25% of them will be red, 50% will be pink, and 25% will be white. So if we do this Punnett square, this is what we end up with. And it's those same ratios. Notice that 1 to 2 to 1 in the genotype is the same as the 1 to 2 to 1 in the phenotype. In codominance, that means that both alleles are dominant. So for example, your blood type. And there are three versions, A, B, and O. So we said in class, or in one of the classes, that basically blood type can be determined by an I. The A and B alleles are both codominant, meaning that both A and B's are dominant over the I and can affect what the blood cell actually, what type of blood you have. So the genes code for a different sugar on the surface, and it's basically like this little tag that will say if you are either type A or type B or AB. So here on this chart, if you look at the left, this is the phenotype. If you have blood type A, then your genotype can either be an I designated with a big A, or an A with nothing. This means if you are type A, that means you are anti-B. And if you follow down the chart, B would be anti-A. AB is codominant, so it has an A allele and a B allele, which basically allows you to accept either blood type. O is the recessive. It is both anti-A and anti-B. So if you have a blood type O, then you can only accept blood type O for transfusions. If you are AB, you are not either anti-A or anti-B, so you can accept any blood type.